Und wir suchen jetzt natürlich ab 115. Ist dann noch auf unserer Liste, das war's. Ciao.
Ist ja gefährlich, ne? Ach, vielleicht läuft es zwar, ne?
Even the addition, can you? Kenny?
Okay. Good morning, everyone.
Morning. Oh, Yojiaka.
Yeah. Yeah. It's not funny here. I'm fine. Are you? Yeah. So I think already we are 14 and we have to move on now. I will share some slides and we shall start from there. Okay. So confirm that we are going to make use of the chat box, and yeah, so you can ask your question. Don't direct it to me. Just make it general. So there are other resource persons here that will attend to that. Okay, so you can use the chat box to direct this to us, and Innocent Mary is here. Our main resource person is also here. So yeah, so definitely it will happen. Then those on site, Vincent will also come around and hopefully can give you some physical attention if you need any. Yeah, so what you're seeing currently is the program for this our meeting that is going to start. It has already started, and through the 3. So of course the 1 is today. So we have a lot to do today because there are preambles and some preliminary things we shall do together before we'll kickstart. Then thereafter we shall look at other things. So we should start with prayers, and I believe our networks are strong. So I will just request Onochie, to lead us in prayer. Just pray briefly. Then from there we could introduce ourselves and move on from there. Okay, so Onochie, thank you.
Okay, in Jesus' name, Father Lord, we thank you. We thank you for this morning. We thank you for our lives. We commit this workshop into your able hands. Help us to learn maximally. In Jesus' mighty name we pray.
Amen.
Amen.
Amen. So thank you. Right away, we shall, just as some persons have done, please could you also make your profile name your complete name. So I like Chikotele Anu James or Kafani Anyale. Onoche Chimamaka. Clear names, full names. So this will help us. Then Bankole who, Amarachi who, what is Adie who. So let's reflect our full names in the stuff. Okay? Yeah, Chokwode Michael which is okay. Noelon Vivian, fine. Princess Sonia Obona, fine. So let's reflect our full names in the stuff. That will help us. And maybe when you are introducing yourself, you can turn your video. Then we shall see you just as you're seeing me. Then we could now sequentially proceed like that. Then after you must have introduced yourself, you can off your video again until maybe when we request for that. Okay? Yeah. Okay, so we shall start the introduction of participants from the lady that led us in prayer. Then if I mention your name, you can on your video introduce yourself. So name and maybe your department or what you're currently researching on, and maybe why you are in this training and what you hopefully wish to gain at the end of the training. So I think just, this ends in 12 minutes time. So yeah, Onoche, you can go ahead, Mrs.
Okay. I am a pharmacist, Ifeanyi Onochie, in the Department of Biotech and Microbiology, Nnamdi Azikiwe University. Then I'm working on the prevalence of dengue in HIV patients. Then, well, why I joined I just wanted to learn. That's why I joined the workshop, yes, to learn more, yeah.
Thank you.
Thank you.
Chikodli Anugu. So we move over to Chikodli Anugu. James or Kafanyale, if you're there, you can. Is Chikodli here?
All right, James, I'm here.
Yeah, go ahead.
Okay, Chico, you can go ahead, please.
James, you can go ahead. Chikodli maybe not there. Michael, you can go ahead.
Okay, good morning, everybody. Hello, can you hear me?
Yeah.
Good morning. My name is Dr. Chukwu Michael Ebuchi. I'm in the Department of Parasitology and Entomology. I work on malaria parasites and vectors. On the aspect of vectors, I look at the insecticide resistance status of Anopheles mosquitoes. Then on the aspect of the parasite, I look at malaria parasite diagnosis using different tools. So I'm interested in bioinformatics and this workshop as well, so that I can improve the scope of my research. Thank you.
Thank you. Yeah. James, are you ready now?
All right, thank you. My name is James. My name is James Okorafor from Biotech Research Center.
Yes.
Yeah, from Biotech Research Center. I currently don't.
James, are you sitting there with someone else that is doing this training? Huh? Okay, thank you, James. Could we move over to Noyalum Vivian? And so Noyalum Vivian, I will unmute you for the introduction. So Noyalum Vivian, you can unmute now.
Vivian is present.
Go ahead, please introduce yourself.
Okay. Good morning, everyone. Vivian Naelo Manakwenze, it's my name, from the Department of Applied Microbiology and Brewing. We have a work we are doing on this fever, and I hope that knowledge gained from this workshop in the research. Thank you.
Okay. Adia Francisca.
Hello.
Yeah, you can put your picture on as you introduce yourself.
Okay, good morning. Good morning. Can you hear me?
Yeah.
Okay, I'm Adia Francisca from the Department of Microbiology and Public Health, Cross River University of Technology, Calabar. Okay, I think knowledge, I'm working on antibiotic resistance in agricultural environment. I'm interested in learning, that's why I applied for this, and I think I'm interested in bioinformatics too. That's why I did this. Knowledge gained from here will help me a lot in my research work. Thank you.
Okay, thank you. That's nice. Okay. We move over to Inosa Benjamin. Inosa Benjamin.
Good morning. Hello.
Yeah.
Hello. Good morning. Are you hearing me, sir?
Yeah.
I'm Inusa Benjamin from Molecular Research Foundation in Oka University, Nnamdi Ezekwe University. I did my master in pharmaceutical chemistry, abuse area, and I'm also interested in bioinformatics and chemoinformatics. Thank you.
Yeah, thank you, Inosa. Is Lillian Chidi Honora?
Yes, I'm here.
Yeah.
Lilian is here.
Lillian, could you introduce yourself? We are doing participants introduction.
Okay. Lilian Choma Chidi Honora Mrs. I want to participate in molecular docking as to acquire more knowledge to assist me in my PhD research, which is still ongoing in drug resistance in the Aspergillus species. So acquiring knowledge from this will go a long way in assisting me in my research. I'm in applied microbiology and brewing, Nnamdi Ezekwe University, Oka. Thank you.
Okay. Thank you. It's Andrew Kuchi Godley. Could you?
Yes, Anao Kuchi Godley by name, from the Department of Applied Microbiology and Brewing. Yeah, my research interest or area of research is looking at polluted environments, trying to understand the biodiversity and abundances, of course, employing metagenomics, so as to understand functional genes of the microbial populations in the polluted environments. Thank you.
Yeah, could we ask Marachi Cynthia Akachubu? Akachubu Amarachi. Princess Sonia, could you introduce yourself? Then while doing that, we request for the video to see who is participating with us. So Princess, if you're there, please, Sonia Obonna.
Yeah, Sonia is here.
Yeah, so you can on your video and introduce yourself. Unmute yourself. Okay.
Okay.
Yeah. Sonia? So is Joy Ginika here? You could introduce yourself. I've requested that you unmute yourself.
Okay, hello.
Who is talking to us?
It's Sonia.
Okay.
Okay, it's Sonia Obona from Department of Microbiology.
On your video.
Okay.
Bankole, are you here? Bankole, are you here?
Yeah, I'm here.
Yeah, you can introduce yourself, please.
Okay, good morning, everyone.
Huh?
Can you hear me?
Yes.
Okay, so I'm saying good morning to everyone. And it's a pleasure to be in the workshop today. My name is Bankole Ogbashire. I'm a med lab scientist. I work in a research lab in Lagos here. We carry out procedures such as whole genome sequencing, whole genome genotyping, and so on. So I believe that one of what interests me in joining this bioinformatics training is because I believe that it's actually a major part of whole genome sequencing procedure, part of the result analysis and the like. So looking forward to learning, having a great time in the workshop. Thank you.
Okay, thank you, Bankole. It's Atanda Halimat here. Atanda Halimat. Engineer Christian.
That is Dennis Aribodo using this.
Okay, Professor, you can go ahead.
Okay. Am I being seen?
Yes.
My name is Dennis Aribodo, Professor of Public Health Parasitology, Nnamdi Azikiwe University. I have a bias for malaria parasites. Thank you.
Thank you, Prof. Ihe Kurume, Chidioka, are you here? Ihe Kurume, Chidioka, are you here? Wow. Yeah?
I'm here.
Could you introduce yourself?
Okay.
For video to be on, if that is possible for your end, but it's important to know people we are working with. Yeah. Go ahead.
Mrs. Marigold Ihukumere from the Department of Applied Microbiology and Brewery, UNIZIC.
Okay.
I'm interested in the bioinformatics and the molecular docking so that it will help me in my PhD research project. Thank you.
Thank you. Okay. Is Joy Gineka here now?
Good morning, everybody. Good morning. Can you hear me?
Yes, could you also on your video for the purpose of introduction?
Good morning. Good morning, everybody.
Good morning.
Okay, I'm
Yeah,
Joy.
good morning, Joy. Can hear you. Go ahead.
All right, I'm Joy Ginika. I decided to join this molecular docking workshop in order to help me in my research. Thank you very much.
Thank you. Finally now, is Amarachi Cynthia here now? Okay, so quickly I will introduce the resource persons that we have here today, or I will call them, they will introduce themselves, who are going to work very closely with us to ensure that this workshop flows and flows well. One of them is a pharmacist, Innocent Mary, who already is assisting several of us, of course. Yeah, I think Miss Ike Okarafor, sorry, could you introduce yourself now? And maybe Sonia, if everything is fine. Ike Okarafor.
Okay, I'm sorry, I'm still working with my video. Thank you.
Is Princess Sonia ready?
Yes, I'm here. Hello, I'm here.
Kindly introduce yourself. Let's make progress, please.
Okay, I'm Sonia Obonna. I'm a lecturer in applied microbiology and brewery. I'm interested in the docking because it's going to help me in my PhD work, and I'm glad to be part of this. Thank you.
Okay. Then Ike Okarafor, quickly.
All right, my name is Ike Okorafor, and I happen to work at Evangel University. I'm interested in the molecular docking workshop because
this link.
it's going to help in my work, and I'm also curious about what it's all about. Thank you very much.
Yes.
Thank you. You get satisfied today. I think we have taken everything. Halimat.
Hello.
You introduce yourself. If the video is okay, you can put it on and introduce yourself.
Good morning, everyone. My name is Halima Atunta. I'm a biotechnology student of the Federal University of Technology, Akure. Okay.
Yeah.
I saw the program on the Telegram group and I decided to join because I'm interested in bioinformatics, so I'm trying to expand my knowledge.
Okay. Thank you, Halimat. We follow. Yeah. I think we are left with one person, Amaka Obianom and Akajuku. So if they can, I think they should go on to introduce themselves.
Okay. I'm driving, I'm coming. That man is there later. Network, can't even hear it.
Okay.
you hear it?
Yeah. Okay, so I think with this, it's okay with the introduction. Maybe later we could still take some that are not yet introduced today. So next in our program is to welcome all of us, but I'm going to now do that with the program overview. So of course, we are going to focus on molecular docking in these 3 days. And this program is 4 hours per day. So we start 11 and we end 2 p.m. Okay? That does not mean that you will end 2 p.m., but officially we could close, but your hands-on and your practice could continue wherever you are. Okay? So we have few things we want to achieve, and we hope and trust and believe that it will work out fine. Okay, number one is to introduce you to bioinformatics again. I use the word again because I know you already been introduced, so we shall not waste much time on that. But it's still part of our program because there are some persons that this is their very first time, to participate in a bioinformatics training. So we shall do that. Okay, then immediately after that, we have 2 things we must achieve today, which is biological databases, and also chemical databases. Then immediately we shall also do hands-on section on these 2 databases. Of course, I've sent the hands-on section guide to all of us in our email. So I hope that you will be able to go through it and flow along with us. Okay, so once again, I welcome you, and maybe I will do more of that as we take a look at the introduction of bioinformatics. Okay, so we shall quickly introduce bioinformatics to us. So maybe you could confirm through the chat that you can see the slide. So if you can see through the slide, please, you can write to everybody, not to me, and say, yes, I can see this, or I can see. So once you confirm that, then I could start. Okay. Thank you. To confirm that you can see everyone. Okay. Thank you. Okay. Yeah, thank you. But before I go ahead, it's because I'm getting not really bored, but maybe fluctuations in the internet could not allow the introduction to flow as I wished. But I will not also forget to ask our resource persons in the room to introduce themselves. So Innocent Mary, maybe you can do this for yourself since your network is relatively stable. Is Innocent Mary here? Yeah, we also have we also have Dr. Samuel. We also have Dr. Samuel.
Hello. Amaka Obianom is here.
Who?
Obiano Mamaka.
Okay, can we just get your introduction quickly because we're already moving on?
Oh, sorry about that. Good morning, everyone. My name is Obiano Mamaka from the Department of Applied Microbiology and Brewing. I'm excited to be here today, and I want to learn, and I'm interested in the workshop on docking. Thank you.
Okay, thank you. Dr. Sam here.
Hello, sir.
Okay, so yeah, I wanted you to do your introduction as
Oh, okay. My name is Samuel Egeye. I'm a senior lecturer at the University of the Western Cape, and I am glad to be part of this. And hopefully, by the grace of God, we'll be able to share together and learn from each other. God bless you.
Okay, thank you, Sam. I don't think Innocent Mary is on seat as, yeah, so I will go on because after now I will hand over to Dr. Sam, then who will guide most of the other things we shall do today. So, yeah, so I am Moses Ikechukwu, preferably, though my surname is Ikebunam, yeah, officially. Yeah, but my name you could call is Moses Ikechukwu, yeah. So first, I have 2 affiliations, or even 3 affiliations currently. Number 1 is the Department of Pharmaceutical Microbiology and Biotechnology, Nnamdi Azikiwe University. Yeah, that was my affiliation originally, and they stay counting. Then secondly, I am currently affiliated with Institute for Tropical Medicine, Tübingen, Germany. So where I have been doing my postdoc fellowship for, yeah, going to 18 months now. Yeah, so here currently we don't do bioinformatics as per bioinformatics, but a lot of deeper molecular biology and the expression of vaccine candidate antigens of malaria parasites. So, and also to optimize PCR for drug resistance assays. Yeah. So I also have affiliation with Molecular Research Foundation, who is also a member of the NGBC Consortium. She is a member, yeah. So I have 3. Okay. So today I'm going to introduce bioinformatics. So for those who have never been to bioinformatics workshop, then maybe we could be a little bit gradual with you so that you could also flow along. Yeah, part of the things we are doing currently is to sensitize people a lot on this new area of research. And once you're sensitized and you have interest, then you could drive the entire course yourself. So we never fail to continue to introduce bioinformatics. Maybe in the course of the introduction, you will get to know many other possibilities, many other research areas that you could also flow along. Yeah. So what we're doing today as a training docking, you'll also see where it falls into. Yeah, but there are many areas in bioinformatics, and maybe in the course of this introduction, you will get to know. So I will quickly talk about what bioinformatics is all about, then a little definition, essence, milestone, and the why bioinformatics, and the resources for bioinformatics. Then I will mention some data basics, even though Sam will in detail through his videos and interaction with us, do more on the biological databases and the chemical, but I'll just do an overview of the whole thing. Then I will also mention some applications of these and share my conclusion finally with us. Informatics. So maybe you could have a very clear picture on, I don't know, okay, I think, let me use a laser pointer. Yeah, so you could have a look at this picture here. From here, you may begin to understand and conceptualize what bioinformatics is all about. This is a course that have attracted many disciplines. And with what is going on currently in research, you must have an application of bioinformatics in what you're doing. Yeah, in one way or the other. You may not be a bioinformatician, but you could apply bioinformatics in what you are doing. And a lot of researches going on have shown that bioinformatics has brought many disciplines together. Okay, so but the pro issue is what it is. What is bioinformatics? What is bioinformatics in its sense of definition? So we could say that bioinformatics is, sorry, I dropped this pen for something. Yeah. So I could say that bioinformatics is the application of computer science, yeah, including physics, chemistry, biology, molecular biology, biotechnology, etc., because we shall not be able to outline all of them. So application of all of these on biological data for better data organization, for easy submission of data, for retrieval, updating and manipulation, as well as data analysis. So, and you could see that there is no how one person will be able to do all of these. So you could decide your own focus and move on with it. So first, again, or secondly, that bioinformatics is multidisciplinary and is building bridges to connect all research discipline in life science. And the ultimate goal of this is to acquire new knowledge from already existing biological data that technology is generating to improve the quality of human life and other forms of life. So this is very, very important. Yeah, so bioinformatics it's already working on already existing data. So in the course of this workshop, you discover that the proteins, the ligands, some of the things you are going to work has already been deposited by some persons that worked on the wet lab to elucidate the protein structure. So, but your responsibility now is to now use the protein deposited to apply it in your own area. Maybe you are in agriculture and there are plant diseases, so you are in plant pathology. So plant diseases, and you really want to look for compounds that will be able to handle some plant diseases. So this is also an opportunity to look at plant database and also some proteins that the organism or causative organisms are causing, and they do the docking and make some interpretation and now do the in vitro and finalize, as the case may be. So bioinformatics cuts across every field. So those in computer science, engineering, physics, and everybody is there. So in one of the meetings we attended last 2 years in UK, I met a physicist that is a molecular biologist. So I cannot explain this, but that is what bioinformatics could do. So no matter your background, where you're coming from, you can make your career in this discipline. So there are many flavors of biological data. Just as we know in, what do we call in our research, we could say biodata of someone, name, surname, age, the region is coming from, religion. So when you get all this information about someone, you could call it the biodata of the person. And maybe you can put it somewhere in the computer. Okay? So then data is beyond biodata. And And the data could be DNA sequences. Data could be amino acid sequences. Data could be protein in 2-dimensional structure. Data could be protein in 3-dimensional structure. So this is where you could find all the 3-dimensional structure of a protein. Data could be in form of images. Then data could be in form of expression data sets. That is, you're talking about RNA and the messenger RNA. It could come in form of libraries. It could come in form of tags, those that have been tagged, those that have been curated, that is those that function, that has been assigned function, and the non-curated, those that, of course, their function has not yet been known. So all of these are different data flavors that we could have. In this, our molecular training, we shall look more on protein database, which is either in 2-dimensional structure or in 3-dimensional structure. Those who have been attending our trainings in the past will discover that we have done much on DNA sequence database, gene bank, NCBI, and all of that. So in this training is more of protein structure, amino acid. So we want to go where it is. And every human being, every animal, every parasite, every malaria parasite expresses protein. So what shall we do with this protein? Where do we get this protein? So we shall secure this protein in protein data bank. So this is where they store all the data that has to do with protein. So I'm saying this in this way so that we can refresh and connect back and approach the training with focus. Okay. So bioinformatics, of course, constituted by databases, tools, or algorithms. Algorithms has to do with the flow, workflows, and all of that solves a particular problem. Personnel and network to bring about knowledge output from biological data. So all of these things happens, So bioinformatics is now a hub and a turning point for modern day research and development. I will tell you more. Currently, a lot has happened in COVID, and without bioinformatics, we wouldn't even know the variants of the COVID-19. But it's genomic sequencing, it's genomic application of bioinformatics that revealed that UK had their own peculiar variants, and almighty Nigeria also have their own, special variants. It's bioinformatics that made this possible. So all of this bioinformatics, okay, let's not preempt issues. Let's go ahead. So you could look at the hub and you will see that bioinformatics cut across all of these, computer-aided drug design, which is what we are going to flow a little into. Molecular biology, those that isolate DNA, sequence DNA, and make the sequences available so that someone can use his computer to interpret the sequences. Such person we call a bioinformatician. So some also work on genomic, proteomics, transcriptomics, or the omics in the wet lab, release the data online, and the bioinformatician, we do what? We use the computer, analyze the genomic data. So bioinformatics is a now a turning point across many. So those in the IT, they develop software. That is why Anedu is with us, who is a computer man. So we develop software because gradually in NGBC, we should have peculiar databases in UNIZIC where many data of some of the bioinformatics that have been done are assembled together and people could retrieve this and compare with what they're doing. Yeah, so that is it. So I mean that this is an interesting field that each time, even though for some time, I am more of wet lab now, doing a lot of molecular biology. But each time we have to talk about bioinformatics, I must say that I am interested, deeply interested. Yeah, I still apply here to do basic DNA sequencing, analysis and all of that. But these are basic bioinformatics anyway. So I hope you will find your place in this as we build on. So in bioinformatics, biology interacts with informatics to give birth to genomics, proteomics, transcriptomics, metabolomics, pharmacogenomics, etc. So there is coming together of biology and informatics, to give birth to all of these things. And I wish that you take decisions on what to do with your laptop hence, whether it's genomics or proteomics or transcriptomics or metabolomics, and go into it, seek for more training in that area and build on from there. So bioinformatics is a key area. Number one is the sequence analysis. And this is my own interest. This is my own area. Yeah, I applied it during my PhD to study single nucleotide polymorphisms of malaria-resistant genes. So that was, I assessed all the drug resistance against almost all the malaria drugs, though I still had affiliation with my current lab here during that time, so it was possible to do a lot without spending a lot as well. So sequence analysis, so you work more with a molecular biologist who does the DNA amplification, who does the genomic sequencing, and deposits the sequence in a database. So within the database, you can now source. When I was doing the single nucleotide polymorphism for artemisinin resistance, which I amplified a gene known as KH13. So in that particular analysis, I needed to go to the PlasmoDB. PlasmoDB is a database dedicated to Plasmodium species. Okay, dedicated to Plasmodium species. So from here, I secured what they call the reference sequence, with which I was able to dictate my own samples that had mutation at a particular codon. This is very important. So sequence analysis is an area, then those who are also in databases an area, then structural bioinformatics, molecular docking, which we are going to study today, is basically on the structural bioinformatics. What do we mean by that? The central dogma of life reveals to us that it is already basic that life is coded in the DNA. And this DNA is transcribed into messenger RNA. And from this messenger RNA, this moves on to protein. So you can understand protein from DNA. And you are who you are because of your protein. You are tall, you are short, your eyes can see well. Actually, as you get older, begin to have eye issues and the ocular matters, then it's because, of course, the protein expression is gradually reducing so that you can leave the earth for others to come. So that is the way it is. So in structural, it's more of protein studies. Protein studies, an interesting area. I got so much interested in all the areas that I can say as focus, I'm more in sequence analysis, but we have papers in structural bioinformatics already, just interest. And all of us in this training, I want to say to you that it is possible after this training, you could tell your students to buy only laptop and internet for their undergraduate project. Because there are a lot of questions, all the candidates that they are culturally in microbiology, we can begin to now seek for which drug can enhance and inhibit candidate through bioinformatics. So these are possible, and maybe as we interact, we could still see. So in sequence analysis, there is homology searches. Okay. There is homologous starches. Then the databases is where the data are organized. If they are not organized, you cannot access them. The terms of sequences, structures, and the functional data are organized there, and we could easily source them based on that. So in the structural bioinformatics, just as I've already explained, from the sequences, you can, of course, flow to the structure. You can also predict function. You can also predict structure. So the whole thing is there is a tool, of course, a lot of you could keep in the nucleotide sequence, you translate them to amino acid. You could also, from the sequences, predict structure. So, and I know that Dr. Sang will lead us in all of this in this training. So it's going to be interesting if you are if you personally are interested, you know, and be sensitive enough to know an area that you must begin to push personally to get to build yourself and become a resource person in that area in the forthcoming workshop. So our time is running because we have barely 2 hours left. So I will not go into this history. So this is the milestone of many activities that have happened in molecular biology up till the day of bioinformatics. Okay, when the Watson and Crick elucidated the DNA model, the Sanger sequencing that took place, and all of that, polymerase chain reaction that came in 1983, as the case may be, you know. So, but I don't think we have to take our time in history here. You can always get history. Okay, so these are also the origin of bioinformatics, which is very important. How did it start? You know, it was starting gradually, but they never knew that the term bioinformatics, or what they're doing is bioinformatics. Yeah, when the first protein sequence was reported, that was bovine insulin in 1950. Of course, they were able to look at 51 residues. Then nearly a decade later, the first nucleic acid was reported, that of T-alanine. And this has 77 bases. It was a breakthrough in their own time. But you know, currently the entire human genome has been sequenced. Yeah, but we need to start somewhere. And you that is coming into bioinformatics, you also need to know how and what happened and where the story started, and also see the possibility of building also on the story yourself. And this woman is an important personality in bioinformatics because she's the very first person that gathered all the available sequence data to create the first bioinformatics database. Could someone guess how the nature of the first bioinformatics database? It is a book. And this is the book, Atlas of Protein Sequence and Structure, in 1965. And Dayhoff, of course, have joined his fathers, has gone the way of all the earth, you know. But these are the ones that laid the foundation. So the more you can help humanity, please help. The more you can do your own, other people can come. Well, in Nigeria, 2 things will happen. Either they build on this or scatter the one you have done. So, yeah, but do your best for your own generation, and let's move on. Share your knowledge and give people opportunities, and let's see what will happen. So this woman is very important, and if I'm to take a quiz in this course, maybe I will ask you, you know, the very first bioinformatics data we had and in what form it is. But just take notes as we progress. Yeah, so the protein data bank followed in 1972 with the collection of few S-rays, 10 S-ray crystallographic protein structure. Then later in 1987, Swiss protein sequence database began. Yeah, so when we start the database in full, maybe we can also talk about, but much later, sorry, I need to change something that is really interrupting me here. Okay. So the term bioinformatics first appeared in 1970 in Dutch, and then in 1978 in English. And the first graduate of bioinformatics is Japa Heringer in 1984. So in 1982 or three, PCR came. Yeah, so not quite, just a year after the polymerase chain reaction was discovered, then we had the first graduate in bioinformatics. And it's interesting. Okay, so why bioinformatics? Why? It wouldn't have been possible to manage the current data that is coming out from sequencing if not bioinformatics. So why? Because there are a lot of data, that is coming out of the Genome Project. So just as I've said earlier, the Human Genome Project that began in 1990, of course, currently has been completed, even though there is still proposal to look at Africa, because I think in all whatever, it's only one African that they used, without knowing that in Nigeria, they may have many variations of human being and the ethnic groups that fill everywhere. So there's still a proposal to do more in Africa in the human genome, because when you talk about the Human Genome Project, it's more of Igbo samples that were used. So yeah, so the Human Genome Project made everything possible. Then the reading of the chain DNA, which is sequencing. So the sequencing technology empowered the bioinformatics, okay? And for those who are not primarily interested in the human genome, then the point and the good news also is that several pathogens, I work on Plasmodium and also ESBL in terms resistance of bacteria, extended spectrum beta-lactamase producers. So most of this genome has been sequenced, and they are there. They are there for us to work with. Okay? Then bioinformatics also became possible because of the development of internet in the 1990s. So now information you could access instant, then you can exchange and disseminate the biological data, because without these things, it will be very difficult. So what are the resources we need for bioinformatics? You could see in several mails I'm sending across to you, I continue requesting for a good laptop, a good internet, and that is it, that is the lab. So the lab for bioinformatics is a good laptop, and it's first your from 8 gig RAM and above. Okay, then even though the first resources I must say is your personal interest, yeah, very personal interest, which you could also see in the course of the training, that when you are there, you log on and turn off your video and went to market and come back, and so all of those will also show the level of interest you have in the whole thing. Or maybe you are inside a meeting, attending the workshop. So these are issues. So I think we need to have time for what we're interested in doing and all of that. So your personal interest is what will drive this. And maybe as you want to start now, someone have not installed Chimera, that since last week we have been talking about it. So the enthusiastic ones have already installed Chimera. And when they had issues, they announced it quite on time so that they could be assisted. These statements I'm making are very important, because if you're not enthusiastic, you also affect your resource persons. You affect us. We feel like we're wasting our time. And of course, you could know that, we're not looking for money, as we didn't charge you money. Don't consider that. It's just to demonstrate your commitment that we asked for your 2,000. You would have been free. As me, myself, I'm not paying for anything to do this. Okay, so please, these are very important as we introduce this bioinformatics to you. Then also brain power, which we call functional brain. I believe everybody's brain is functional. It's just to activate, engage the brain. Then also internet access. Internet access. Yeah, I will address this. Peculiarly for Nigerians because we have issues. And please do your best. We ask people to come around in arm's sight just to provide an internet and guide. But it might still disappoint, hence we ask for a backup of 4G, probably from MTN, okay, and all of that. So just do your best, find the best internet very close to you. If it's a Tissalat, fine, whatever, but do your best, do your best to pull through what you want to be or your career. Then the another thing we need is software, and many of them are open source, like the Chimera software. There are many softwares, and some of them free, some of them you may need to buy. Okay, primary data available for data analysis. So Oyibo people have done that for us. It's already there. We can go to databases and get this data. I'm going to jot this, what is database and all of that. Maybe Dr. Sam will talk about this so that we could move forward and why this. So I will just leave this because, yeah, so look at some applications of bioinformatics. In a lot, the most common is understanding variants of pathogens, examples as COVID-19. And they use NGS, is the sequencing. I had a webinar from South Africa. Someone made a presentation and we could track through sequencing the flow of COVID from country to country. You know, the variants as they moved, the mathematical model that is also involved in this. So sequencing bioinformatics has done a lot and has come to improve human health. Now it's personalized medicine through pharmacogenomics, where people could even go for their own whole genome sequencing. And from there, they can have understanding of their peculiarity and how they could respond to drug and treatment and all of that. Then, of course, it could be applied in agriculture and in metagenomics, they study in the natural environment. So we also assist in analyzing the metagenomics data in NGBC. If you send in your data, we do our best, to do that without asking for money and all of that. Okay, then in drug development, part of the things we are doing today, molecular docking and molecular dynamics are part of the tools that will guide us in this. So in this particular training, we shall be focusing strongly on drug development. Okay, not really to develop a new drug, but we can use existing drugs and existing compounds to be able to challenge some microorganisms, their protein, or some diseased situation, as the case may be. So this will be our focus for this workshop. Maybe in May or June, we shall focus on NGS, next generation sequencing. Some of us apply to H3 BioNet. They may not give everybody because the space are limited, but we still have our local training on NGS around May or June. We shall still keep in touch with you and you will get to know about this when time comes on that. So in conclusion, I will say bioinformatics has evolved and is still evolving. You can seek to know how it applies to your personal research, attend workshops, hear more about bioinformatics. Please, every month we do a webinar. This month, we had a webinar on applications and essentials of bioinformatics. Next month, 17th March, we shall do another webinar on prospects of bioinformatics. That will help you narrow eventually. So I believe the current workshop will be a starting point for some of us. And yeah. So thank you and the questions. Yeah. Hey guys, I'm through. And maybe you could respond in the chat. How are you now? Before we move to the biological database, which is the main business for the day. Yeah. So we are recording the workshop from here, Onochie. So later we shall still share. So we are recording.
Okay.
Yeah, we are recording from here because it's the host that can do the recording. So, but the record is for us. So immediately after, maybe it goes to our YouTube and our website, and we can access it from there.
Okay.
Okay, so yeah, there's a feedback on the introduction. Yeah. Okay, yeah. Okay, so Dr. Sam, maybe if you're here, is Sam still here? Okay, maybe Innocent, maybe you can introduce yourself. We looked for you, but it has become obvious that you are doing many things now. Okay, so thank you. So could we have feedback on the introduction to the bioinformatics? Is that okay? Should we move ahead? Any question, any concern? Then we can move on. And while Innocent Medri will get ready to kickstart the lecture on biological database. So Innocent, I think the first should be biological database, then
Biological database.
Yeah, could you do, or I also push it from here?
Okay, let me call it up and see. Biological database, okay.
I will enable you so that you can share.
Yeah. Okay. Where is that?
You can share and they start.
Okay, let me. Biological database. This is one. Can you see it?
No.
You can see the screen?
Yes.
Yeah.
Yes.
Okay. I'm starting it.
Could you make it full screen?
Yeah, it will.
Perfect.
Yeah.
But we cannot hear.
Cannot hear.
We cannot hear.
Yeah. Cannot hear. How do I?
A moment.
Okay. Can any of you hear it?
Yes, I can hear you.
I mean, can you hear the video? Can you hear the video?
No, not yet.
Okay.
Let's go. Yeah.
I want to try whether they can hear. Can you hear now?
I have the privilege of presenting some session of this course on molecular docking. Dr. Samuel,
We can hear.
Okay.
I
we can hear.
have the privilege of presenting.
Okay.
My name is Dr. Samuel.
Okay. Let's wait for Dr. Moses. So once he joins, then I will start playing.
Maybe you could stop the sharing. Let me try it from here.
Okay.
Yeah.
Hello?
Yeah. Confirm if you could hear now. Could we hear?
Oh, you need to go and share the sound also.
Okay, I need to share sound.
Yes, it's an option for sharing sound. Hey, that option, yes. I think that option should be in view options or what? There's some option, other options of more options.
In view?
Share sound.
Okay. Yeah.
And chemical databases
Hearing by
in
now.
this particular session. So we'll start with biological databases.
Is that okay?
So just for the avoidance of doubt,
yes, you can hear it.
Kind of define what databases are.
Oh, okay. Please let them, everybody confirm you
Usually
can
they're
hear.
electronic systems where you have an organized
You
collection of
can hear.
structured information
Yes,
or
yes, we
data.
can
Okay,
hear.
so I'm going to stop it and start from the
And
beginning
the
of
electronic
here.
system is usually your computer system. And these days,
So we want to move on to biological databases following our program. And the presenter will be Dr. Samuel. He's with us in this training. Of course, during the hands-on, all of us, we work together. So, but we need to go through these 41 slides together and move on from there.
good afternoon, good evening, or good morning, wherever you are. My name is Dr. Samuel Ayodele Egeye. I have the privilege of presenting some session of this course on molecular docking. I'm a senior lecturer at the University of the Western Cape, and I lead a research group on computational pharmacology and chemoinformatics. Our focus is computational drug design using bioinformatics, chemoinformatics, machine learning, and data science to find druggable targets and compounds that can be designed into drug candidates for infectious and non-infectious diseases.
Volume, please.
So in this course, I will be taking you through molecular docking, but there will be some sort of a prerequisite a conversation we're going to have prior to that in terms of looking at biological databases. Chemical databases will also look at molecular visualization and manipulation using different software packages. Then we'll now delve into molecular docking. Okay, so I'm going to start by looking at biological databases and chemical databases in this particular session. So we'll start with biological databases. So just for the avoidance of doubt, kind of define what databases are. So usually they're electronic systems where you have an organized collection of structured information or data, and the electronic system is usually your computer system. And these days, the structure of databases has significantly improved from what we used to be, where you know have information in different fields, in different servers, now being pulled together into a central server, and that central server, once a query comes through the interface by the user, is able to go to that central servers, which links to all the servers where the informations are stored, and give an output to the user. And so significantly, the structure around the connection between the individual servers and the central server has improved. We used to have relational database system pioneered by Oracle, but now we have what we call it graph-based system that is a bit more intuitive, and the connection and the network is a bit more complex than you have in relational databases. And the visualization of the data rather that you see once you make a query is significantly better these days. Okay, so you can get more information if you really want to, if you're interested in looking at the structure of databases. But if we now move to biological database, if we want to define that based on the definition we've given to databases, then we can say that it's an organized collection of structured life science information or data that are collected from scientific experiments, published literatures, some high throughput experiments, sometimes from industry and academic research. So even sometimes some computer simulation that have been done by other people. Okay, so all this data are put together and organized and structured in such a way that the user can query and get what they want. And sometimes the database goes ahead to not only give you what you want, interpret the results that you get from such a query. Okay, and the reason why, let me put it this way, the emergence of biological data is actually a response to large amount of data, biological data that have been generated especially from DNA sequencing. Okay, just let me check something before I move on here to be sure that. All right, so what kind of information do we have in these databases? Excuse me. So I'll just quickly remind you of the central dogma of molecular biology, where we know that the DNA or the gene, the DNA is transcribed into the RNA, and the RNA is translated into proteins, and you have proteins that are involved in biochemical pathways. And of course, this eventually leads to the phenotype of the things that we observe in the lab. Okay, so most research in this field try to draw a correlation between or amongst the phenotypic observations and these other path that or processes that take place within the cell, referring to the genomic, the transcriptomics, the proteomics, and metabolomics processes that, of course, we are known now are responsible for some of the phenotypes that we observe in some of our experiments. Okay, and so if we have a lot of information on this and all these processes, we are able to give better explanation to the biological phenotypes that we see. All right, so genome, in terms of specific information in this genomic, which we're going to be like focus on a bit, will be your gene ID, the gene sequences, the gene expression levels, the function of the gene, the structure of the gene, and the rest of them, and the proteomic information, information specifically looking at the protein, the name of the protein, the gene that is responsible for that expressed protein, the function of that protein, the location of that protein, and how the protein interacts with other protein in the biochemical pathway or signaling pathway within the cell. the expression level, the domains, and the rest of them. So this typically just shows you the kind of information that are available within biological database. That it's by no means exhaustive, and you will see more as you get into your hands-on activities that you will be doing in each of this after I finish this series of conversation with you. So if we want to say what are the types of databases that are available, we can generally classify into sequence databases, structured databases, and let's call the others and put the rest of the databases on under others. So according to this author, so sequence databases, it makes sense because you have your nucleic acid sequences, your protein sequences, and there are databases that store this information. They might not be limited to that, but they are basically set out to store information related to the sequences of nucleic acid, and like SwissProt set up to store information related to protein sequences. So you also have structured databases that naturally now, after the translation, able to scientists have resolved the structure of some of these macromolecules and proteins, and they are able to now deposit the structures of those proteins in a particular database, like your protein data bank, and the other refers to like functional, or what we call a secondary databases that take information from this kind of infer functions from either from the sequence on the structure or analyze the structure to give us better information. So you now have, for example, I think I have something on the next slide that talks about that. So you have nucleotide databases like your gene bank, protein sequence database like your UniProt, your structure database like your PDB, protein data bank, and you have what we call the domain or motif databases that actually now look at the structures themselves and try to define, domains within that particular structure. So you have your gene, like I said, gene bank, nucleotide sequences, you have your protein sequences, you have your protein structure databases, and you have some other databases that also explain parts of that particular protein in terms of looking at specific domains and motifs. So domain is essentially a conserved part of the protein that can perform its function or move independent of the other parts. So this is another list that I just made in terms of biological databases. Once again, it's by no means exhaustive. There's quite a large number of databases. So for a specific research area or interest, I can guarantee that you will find a database that has curated and annotated information from different sources and put in that particular database. Okay, just to mention at this point, if you do an experiment and you're confident of the data that you have, you can actually deposit your data in some of these databases. There's a process in which the owners or the people that maintain these databases, when they get such information from researchers, they actually take them through quality control checks, and in terms of really checking the experiment behind that particular data, now try to now explain what the data is in terms of the process of annotating that data, what are the different parts of that data, what do they mean, and then putting an identifier on that particular different aspect of that particular data, and now curate them, trying to put them in specific themes, and in order to make it easy to query, so that people, if they're looking for gene related to, for example, to viruses, you should be able to see any information or research data from that under that particular theme, as it were, or particular structure that they have, or architecture that they've built for that particular database. Okay, so why do we need them? Why is it important? Why don't we just go to Google and type in your thing and say, okay, give me the gene for virus, give me the protein for virus. Okay, give me the biochemicals that are related to virus, to in that particular virus that is, expressed by this particular gene. And, just talking about it is, it's already making me tired, But a biological database it's a one-stop shop that gives you access to a variety of biological relevant data, you know. You can have data ranging from literature, scientific literature, the genome sequence of a particular organism, the gene identifier themselves, and the description of that gene, and the variation in those particular genes, alternate splicing, transcripts, and the rest of them, the structure of protein that result from that gene, biochemical pathways, the chemicals that can modulate that pathway, the disease that results from a particular gene or particular variation. So, the list is beyond this circle that I have here, But what's your biological database? What they bring to the fore in terms of value is that they provide the infrastructure, they provide the architecture for you to be able to have to prepare this database, to manage this database, this particular information, and to mine information from this particular collection of information. So they make it very easy for us to go in there. For example, if you look at this figure, you have this network built by this spider web, in which case basically we're saying that the genome sequence is connected to the taxonomy of that organism, phylogeny, to connected to the biochemical, connected to the disease that is observed from that or a particular phenotype in that particular organism. Okay, so and this network, it's very difficult for you to just collect data and build this kind of connection. It's very, very difficult. So the computer system does that for you based on the architecture behind the specific database that you are querying. Now because these systems are stored within the computer system, this database rather, and the information therein are stored within the computer system, we can do a lot with it in terms of simulation. Doing test mining is a part of your machine learning protocol can be implemented, and sort of other sort of simulations that we can do. Molecular docking itself is a simulation, and you need to engage with the database, get information that you will now use for that simulation. Okay, so it makes it easy for us. We're not just, if the structure of your protein is on the paper, your scientific paper that you printed out, it's not good. It's not good for it to anybody in terms of simulation. But now you have a way where we have transferred that sequence or that structure into a computer readable form. Then we can take that computer readable form and using some other algorithm and begin to engage with that particular structure. Now, the first biological database was GenBank, which is a collection of all available protein and sequences submitted by NIH and NCBI. And the collection of this database actually gave birth to the project of the Human Genome Project, which, because of the advancement in DNA sequences, we are able to sequence the whole genome of a human being. But that was the easier part. The sequencing was the easier part. The most difficult part, the more difficult part was the making sense of the data that came out of the sequence. After doing all the cleaning and calling of the bases and the rest of them. The question now is, you sit with a series of ACTGs in different combinations. What does it mean? Where is the gene? Where is the gene for that particular biochemical process? Where is the gene for that particular phenotype? Where does it start from? Where does it end? Where's the exon? Where's the introns part of that gene? Where are the regulatory part? Where's the promoter region and the rest of them? You need to now account for that, and that is when bioinformatics actually, came in to begin to annotate that particular data. And by annotating them, you can now bring them into a database so that such that if you say, find the gene responsible for angiotensin converting enzyme in humans, you can actually go to that particular chromosome and the loci for that particular gene, and you'll see the starting position and the end position, the promoter region, and the rest for that particular gene. Okay, because somebody has taken time to sit down, not somebody, a group of people. I remember a couple of my supervisors were involved in that project a long time ago, in the South African National Bioinformatics Institute, and yeah, to put that project together. Of course, beyond that, we've done quite a number of things in terms of looking at variations, because I haven't established the genome for humans. We've been able to now come specifically to Africa and say, okay, what does the genome of an African look like? What does the genome of a particular ethnic group in Africa, what does it look like? And that is another information that determines the kind of phenotype we see, why these people are able to get this, susceptible to this disease, and this other set of people are not. Or these people can use this drug, medicine, these other people cannot use this medicine. And we're able to, make sense of that also. So you have a database that goes beyond just the gene bank, looking at genetic variations and the rest of them. So all these databases helps you to, based on the research question that you have, you can always find a starting information that can help you build your hypothesis. Okay. So specific examples of biological databases include all these that I'm trying to list here. You have fungus or Saccharomyces databases, viruses have databases also. I particularly use PlasmoDB because the focus of my research was on malaria. PlasmoDB is part of the VEU PathDB, basically for pathogen, eukaryotic pathogen. And I also use this a lot, protein data bank, which is going to be our focus in this session. UniProt is another, we're going to be looking at this also. It's a comprehensive resource for protein sequences and function. It tries to give free access to sequences of protein and annotate those proteins in terms of the function they perform. And I think it also gives information about the subcellular location of such of those proteins. So it's very important for us to, let me put it this way, that if you do have an understanding of the function of your protein, because we in medicine, different types of biotechnology, expressing, particularly recombinant DNA technology and the rest of them, drug design, and different aspects of biology. We really want to have an insight. We really want to have an insight into the function of a particular protein. So UniProt provides that platform, and is very up to date and a very comprehensive information about proteins. It also helps to not only collect but also interpret sometimes some of this information that are present within that particular database. And it saves researchers countless hours of collecting this information yourself and trying to make sense out of it and everything, and it links to almost over 150 databases. So if you take a protein and you look at the function, you can actually now begin to say, okay, what are the domains with this particular protein? It links you to that particular database that stores information about the domains of protein, which gene actually express this protein. It takes a link and it takes you to the database where the gene for that particular protein is stored, and you can actually begin to look at that gene within the gene browser and the rest of it. Okay, so it's quite a functional, and I found it very useful, especially when it comes to drug designing. structure-based drug designs particularly. Okay, so perhaps this is central for anybody that is doing structure-based drug design. And the central technique to structure-based drug design is molecular docking, which is a focus of our discourse here. Excuse me. Excuse me, I need to set my phone on silent. Okay, so I was saying protein data bank is a three-dimensional structural database of large biomolecule, not just protein, nucleic acid, and some complex systems that are formed either from the interaction of two protein or a protein and nucleic acid, and the rest of them. Okay, but the kind of information you have here are the three-dimensional structures that have been resolved for a particular protein. We know protein usually exists as your tertiary structures. So from your primary polypeptide chain, they form your secondary structure, which is your either a beta sheet or an alpha helix that now folds into your tertiary 3D structures. And of course, you can also have two proteins, 3D proteins interacting to form what we call a quaternary structure. Okay, but the whole idea here is that having access to this 3D structure, for example, if we talk about our focus, this is molecular docking, we want to be able to identify the active sites, or what we call it binding sites, for a particular protein. So we need to be able to, for us to do that kind of simulation, we need that information within the computer system. And this information is basically people who have taken time to use X-ray crystallography or NMR, nucleic magnetic resonance spectroscopy, to resolve the structures of this protein. So what I mean by resolve is basically to account for imagine you have this long chain that is folded into this. You are able to account for if the first part of this chain is alanine 1. The question is, where is alanine 1 in this final structure of the protein? So by accounting for each of this in the final structure, you can build the three-dimensional structure. So you have a coordinate for each of these amino acid that's part of that polypeptide chain in space, where they are. And that is a starting point for us when you want to do your simulation in terms of molecular docking or any other simulation that you are interested in. All right, so I'm going to do a live demo in terms of engaging. I'm going to look at UniProt. I'm also going to look at protein data bank. Then you're going to have an hands-on exercise that you're going to try on your own, and I would suggest that you take notes as you try them. And your on-site and online tutors will also be available to assist you. I'm also available to assist if there are things that you think the online or on-site tutors cannot handle. Okay. All right. So I'll get around to that in a minute.
Yeah, so Mary. Yeah, so we are going to take the online demo immediately. Sorry, I have to. Okay. So biological database to so I share again.
Okay, so this is a demo of biological databases, a follow-up to.
Before we move on to the demo, that is to guide us and to lead us practically how to get a protein, download the protein, and look at Swiss prot as the case may be. Yeah, we shall still send across to you the recorded lecture. He is also here to take questions and all of that. But please, could we feel your concern a little before we go ahead? Should I go ahead with the demo to demonstrate all that has been taught theoretically? Yeah, you can write on the chat so that, yeah, write on the chat. Okay. On the chat, go on. Okay, thank you. Yeah, someone said we should go on. So please, if you are for that, I don't think I have any issue. My major problem was the audio. Go on, okay. Yeah, it's also a function of your own system, but eventually you will still get to have it and use earpiece. Sorry, I forgot to tell us to come with one. I'm using one now, so that is why I could hear clearly. And yeah, so I think we just have to go on with the demo section and move on from there. So please, I'm going on. We have only 17 left here, so.
Biological databases part 1 that I. Okay, so this is a demo of biological databases, a follow-up to biological databases part 1 that we just finished. So we're going to look at UniProt. So a good way to start is to type UniProt on your Google's, and you will see the link to that. Okay, so you have a search bar on top. Once you get there, you can do just a simple search if you know the name of the protein, and just click search and you will get that. There are some other advanced search terms, in which case if you have a couple of terms that you want to combine for your search, that's also possible. I also mentioned the fact, and I can't remember if I did mention, that some of these databases also contain tools that you can use to analyze the information that you get out of them. You have the link to your BLAST. I guess you can check out what BLAST means. It's not covered. I don't want to talk about blasting in this. Alignment of sequences is also possible. Retrieving the ID and mapping it. And you can also search for peptide within this platform. All right. So I'm just going to choose a protein name. You can choose anyone you want in your. And I think I asked for you to choose specific protein in the answer tutorial. So I'm going to use a protein that I know is present in Plasmodium. I'm just going to say search. Okay. So it gives me popular organisms where I can find it, and it gives me a list So the entry here, the table here, that you guess, because you have more than one entry for that FAST13 protein. So there's quite a lot of entry for this one. About 69 entries here. So I can't pick one. I'm looking for the one expressed by the PF3D2, 3D7, this particular gene name. So I can click on that. It's from Plasmodium falciparum to 3D7 isolate. And it tells me the length of the sequence for that particular protein. So 413. Okay, if I click on that, now it takes me to the page for that particular protein. And so we start by looking at the protein name, FAST13, the gene ID for that protein, the organism in which that protein is found, Plasmodium, and the status. Now, like I said, once you put in a particular information, okay, so this is a demo or biological. Okay, so this is a demo or biological databases. A follow-up to the link to that. Okay, so you have a search bar on top. Once you get there, you can do just a simple search if you know the name of the protein, and just click search and you will get that. There are some other advanced search terms, in which case, if you have a couple of terms that you want to combine for your search, that's also possible. I also mentioned the fact, and I can't remember if I did mention, that some of this database also contain tools that you can use to analyze the information that you get out of them. You have the link to your BLAST. I guess you can check out what BLAST means. It's not covered. I don't want to talk about blasting in this. Alignment of sequences is also possible. Retrieving the ID and mapping it. And you can also search for peptide within this platform. All right. So I'm just going to choose a protein name. You can choose anyone you want in your. And I think I asked for you to choose specific protein in the answer tutorial. So I'm going to use a protein that I know is present in Plasmodium. I'm just going to say search. Okay. So it gives me popular organisms where I can find it, and it gives me a list. So the entry here, the table here, that you guess, because you have more than one entry for that FAST13 protein. So there's quite a lot of entry for this one. About 69 entries here. So I can't pick one. I'm looking for the one expressed by the PF3D2, 3D7, this particular gene name. So I can click on that. It's from Plasmodium falciparum to 3D7 isolate. And it tells me the length of the sequence for that particular protein. So 413. Okay, if I click on that, now it takes me to the page for that particular protein. And so we start by looking at the protein name, FAST13, the gene ID for that protein, the organism in which that protein is found, Plasmodium, and the status. Now, like I said, once you put in a particular information, people are allowed to upload data into some of these databases. However, they go through process of reviewing the data that have been deposited, and they kind of annotate that. So you see here, it said it's unreviewed. It means that they are still reviewing that. Once they are able to complete review for this, the annotation score will be high. It's a predicted protein, predicted to be present in that particular organism. You can check out more information on the review state. Okay, now there are major areas. The major areas in UniProt are actually on the tab here by the side. So you have the function, the names and taxonomy, the subcellular location for that protein, the expression, and the processing, molecular processing for that particular protein, in terms of the signal peptide in that protein, and the proteomic databases, especially for those that do proteomic experiments, the structure of that protein, and the family and the domains of that protein. Now, all these are linked to specific databases that actually gives you information on these different aspects that are outlined here. Similar protein also. So this is the sequence of the protein. It also talks about similar protein and references. All right. So if we go back here, you will see the go time for that particular protein that gives you indication or as to the function of the protein. So by go time, I'm making a gene ontology term for that. So you can actually go to the gene ontology page to see. So it's a cysteine type endopeptidase inhibitor. That's the activity of that particular protein. It's involved, regulates the entry of the parasite into the host, particularly the erythrocyte cells. Okay, that's what the biological process is. You can also click on that to get more information, or you go to the complete annotation on Quigo. All right. I can't cover annotation here, but yeah, you can go to the gene ontology and annotation to really get more information about that particular gene or particular protein. All right. So in terms of names and taxonomy, it gives you the name. The gene name is written here. There's a link to that. This is the organism. Taxonomy identifier. You can get the phylogeny information here. So I showed you the VEU part database. Okay, see, there's another link to that database because you have information about this. Full information about the genome of the Plasmodium in this particular database. Then the subcellular location of this protein in the cytosol, in the host cell cytoplasm. Yeah. The domain of the protein and the sequence of the protein. So what is what we call a FASTA sequence, which you can download. The FASTA sequence. As a matter of fact, if you click on it, it gives you this document. You can actually copy this and just put it on in a text file and save it as FASTA for you to do some other things. All right, cool. But what I actually want to, there's similar proteins and the references. But what I want to focus on, based on what we're going to do, is the 3D structure. So usually, if the structure of this has been resolved, you will have a PDB ID for this particular protein. So this is basically what I'm saying is this. So if I go to another UniProt site and I type in, I'm trying to find the protein. Okay, I'm just going to call it ACE2. which is your angiotensin converting enzyme 2, which is the receptor for coronavirus. Yeah, in the human. If I click on that, so angiotensin converting enzyme 2, ACE2, gene name present in Homo sapien human, and outlines the function here. But this is where I'm going in terms of, so if you go to this tab, you can actually click the tab and it takes you straight to the place, the structure. Okay, so this is what you see. This is actually the structure of this particular protein. Okay. So you don't have that here because if you go to the structure, you don't have it here because the structure is not available in the protein data bank. Yeah, so in this case, so many people have worked on this protein and deposited the structure in the protein data bank. Okay, yeah, so you can actually go to any link from here to actually go and see the structure. And you see, like I said, the databases also provide some tools for you. They have a visualization tools here for you to look at that particular protein. All right. But for if you don't have that structure in protein data bank, there's some other databases that, what's the word I'm looking for, that try to model the structure. So you have ModeBase, which is a site that tries to store. Some people have said, okay, fine, we can, based on similarity to other protein, model the structure of this particular protein, even though we have not experimentally established the structure. We can build a model from the knowledge we have from other databases. So you can have that where somebody has actually build a model. This is what you have. Somebody went to build a model for that particular protein. Yeah, and this is what it looks like. Okay. So you if you really need to work on this, you're not limited. You can actually go and get the structure from that particular site and begin to work with what you have, rather than saying, okay, there's no structure, and that's not a limitation. The other option will be to, if you go back to the sequence, which is here, you can take the sequence, download the first file, and take it to some other platforms that can actually build the model based on the sequence of that particular protein. Okay, so that is also very possible. All right. So just once remember, you will definitely have some that have structures in protein data bank will be shown here. Yeah. And you can select any one of them you want. Look at the secondary structures also shown here. Applying the helix, the turn, the beta strand, yeah, of the protein. All right. So I'm going to, so you're going to play around with UniProt and you're going to use it to get some things. Okay. So let me stop there for now. Then we'll go to protein data bank, which I think is more of what we want.
Okay, so yeah, I'm excited with some of the things I'm seeing here again. And yeah, so we're going to move on the third biological database to look at the protein data bank in details. Then thereafter, we should have our hands-on, because you have to go to the UniProt yourself, get the protein for our hands-on, download the proteins, and we shall move on from there. So I think I can stop this to start another one. Yeah, biological 3. Okay.
Okay, so the last session we looked at the UniProt, and one of the things that I showed was this protein, angiotensin converting enzyme 2. And my point was that it has a structure that has been solved. Okay, and on this, on the right hand side, you'll see the different, well, not is the structure of this same protein that have been deposited on the protein data bank. Okay, let me just take it. You'll see that they have specific, what we call a PBD entry, specific ID, and the method by which the structure was solved. See stratigraphy. This was a model built computationally. There's also electron microscope as a way to solve the structure as a method. Then you have the resolution of the solved structure. The higher the resolution, the more confident you are that the experiment actually got the coordinates of the amino acid, like I spoke about, the correct coordinates of the amino acid in the final 3D structure. So you want to pick the highest coordinate. The X-ray structures is okay, but modern strategies of using electron microscope, so you can also explore the structure that are built from that. Then the chain that is in the protein, sometimes they might just model or resolve one chain. The chain refers to the fact that, like I said, a protein can have two different 3D structure that comes together in a dimer or tetramer or something like that. So sometimes you might have they are able to resolve both major chains involved in that protein, A and B. Some just did the A part of it. Yeah. So depends on what the experiment that was done and which part of the structure was resolved. Some have everything, different aspects or different chains that constitute that particular protein. All right. But yeah, so you can actually now, and this is the position in terms of the length of the protein, in terms of sequence length, and finally links to where you can get them. So you have the PDB Europe, PDB RSSB, maintaining the US. I think what was there again? Look further. There we go. Research Collaboratory for Structural Bioinformatics. They also maintain a database to store digital representation of 3D model of protein structures. Okay, where are we? Yeah. And there's still PDB Japan and the summary PDB page. So I think I opened one of them. Okay, this one, the PDB sum gives you like a summary page of the entry, and describes. So in this case, the ACE receptor, ACE2, like I said, is the main receptor for SARS-CoV-2. So the structure was actually resolved together with SARS-CoV-2. Okay, so you can see that's the spike protein of the SARS-CoV-2, and that's your ACE2. All right. Some quality checks are done, which I will show you the details when we get to the BDB. So I'm just going to from this page, like I said, you can get straight from the UniProt with a link to PDB. But assume that you went straight to PDB. So let's say you went straight to, so I'm going to go back to Google. If you can just type it straight if you want to. And I'm going to say, if you write PDB, I think in my tab, it takes me to the RCSB PDB homepage, and that's most frequently PDB page that I use. Okay, so you can type it straight here, or you go straight to the home page, and now then on the home page you have the description and some news about molecules of the month, recent submissions, and the rest of them. Okay, but this is where your interest is. So I can type in the angiotensin converting enzyme 2. All right, so it gives me quite a number of options, but I'm just interested in that. I don't want it linked to, yeah. So most of the entries, because of the recent coronavirus, have the ACE linked to the spike protein of the SARS-CoV-2. Okay. Most of them have that. So these are the different entries that have been submitted to put into the bank. Now, this one is the native human angiotensin converting enzyme related captosepeptidase. It's linked to a particular peptidase. Okay, so it might be an interesting one to look. So this one also has an inhibitor attached to it. So let's take a look at this one. There might be some interesting things to see. Okay. Okay, so you have a description of the protein itself. It's a hydrolase, Homo sapien, the organism, where it is located, where it was found. It was expressed in this system before the resolution was done. Okay, that's the date it was deposited. Of course, it was checked and curated, released on this particular day. You have the link to the paper of the guys that actually worked on the resolution. All right, so just here, I just want to show you that each resolved structure is analyzed, and there's a quality check done using different parameters, and they are scored based on their performance, based on that particular, on each of those parameters. Here we have something called a pro check on that PDB summary. And one of the things that is there is your Ramachandra plot. We can check that out. It's a way of checking if the amino acid are all in the appropriate position that is allowable based on the bond that they carry or share with other atoms in that protein. And you can just check it out. But here they give this metric a R-free class score, Ramachandra outliers, based on this Ramachandra plot that I showed you, side chain outliers, and the rest of them, and you can go into details, just look at the full report, tells you the quality, all these are metric that tells you the quality of this structure that you're about to work with. And remember, the resolution is also given. Yeah, the higher the resolution, the better it is. All right. Okay, so a couple of things I just want to show you here. The structure itself, you can click on the 3D viewer. So the PDB database also has a visualization tool. allows you to visualize this protein. So if you click on that, it takes you to a visualization tool. I think this is powered by NGL. That powered this visualization, I think. Okay, so yeah, there we go. So with this, you can move the protein around and view the different domains of that protein. So that's your ACE2, and that is the protein that was attached to it, that was bound to it. Maybe there's something about trying to see if this peptide can inhibit this particular protein. Okay. I'll just go back again. What else I want to show you? The ligand interaction. So there's a ligand bound to this. So let's see how they interact. You can also click on that. All right. So this is a ligand that is bound to this protein. And this is you can actually look at the identity of this if you put it. You will see the identity just at the bottom there. If I just point my cursor on that, it's a 2-acetamido 2-deoxybeta glucopyranose. Pyranose is a sugar attached to that protein. So you can zoom in and zoom out. So you can play around with this and just check proteins of your interest, just type. And you can also say a class of particular protein. If I say protease present in HIV virus. So it gives me a couple of those proteases that I can look at. Let me see. This is bound to a particular inhibitor. Just take some time to come up. Here we go. Okay. See the protein with the ligand in it. So that's the ligand. Let's see if we can get the identity of that ligand. Okay, it's not showing here. There are multiple of them here. And if you go closer, you'll see how the ligand is interacting with the residues of the amino acid through these bonds, hydrogen bonds, yeah, that are formed with the residues or the protein. All right. You can also download this. Click on that. You can either download the faster sequence, but in our case, we need the 3D model, so you can download the PDB file, or you want to zip the file or the PDBX, but I would prefer on the platform that we're working, the PDB format should be fine. So you click on that, then you ask you to save it. You click on save. You browse to the folder. I would suggest you have a folder for the training, and it's folder for each session of the training within that same folder. So in this case, you went to the my downloads. Okay, so please, you can actually move it from here to the specific folder that you want. Okay. I think that's about what I'm going to there's a hands-on exercise that we want you to do. Take a look at the PDB and download a particular protein that you're going to use for the docking simulation. First, we're going to just visualize that, then before we do the docking simulation. Okay. So the next section we'll be looking at how you can visualize this macromolecules of proteins and how you can manipulate them as required. I'll talk about that.
Yeah, nice. So maybe I will stop at this point. Then Dr. Sam could take some questions that are already coming in, and we just discuss before we now take the last slot. Maybe not last slot anyway. We still have chemical databases, but let's first review. Let's first review the biological databases, as even myself, I have some questions. So, Doctor, I don't know if you're here. You can on your video and we could have some interaction before we continue.
Yes, sir.
Okay. Okay. Yeah. So this is Dr. Samuel, who has been presenting. So yeah, through the pre-recorded lecture, and he's here also for an online, practical questions. Of course, we have hands-on exercises that we shall all do together. So far, so good. I think you could see the question from Halimat Chisom.
Okay, I need to go to the chat. I'm not sure.
Yeah,
my chat is updated.
Yeah, I think it's updated.
Okay, I'll check.
But though I told her that we have to go on, so that during the hands-on, so she was saying that she can't see the 3D structure under the structure section in UniProt, that she can only see the Swiss model and the mode base.
Okay, she was probably looking at the FAST13. So I had two things open. I had FAST13.
Yeah,
and I had
because I
ACE2.
know that the FAST13 has no 3D structure yet. Whereas the AC2 has already 3D structure.
Yeah.
Okay. So yeah, I think something new came in. Okay. Yeah. So Alema, that is the issue. Yeah. But already, if you could open the hands-on exercise, you begin to also see the particular protein we should use and how to assess this protein. Yeah, for your own use. So I think we could quickly, because our time is running fast, and we have just like an hour so to end the day. Maybe, guys, if everything is okay with biological database so far, because you will still apply this online here, then we could go on to the chemical. Is there any question? Is there anything? Because the lecturer is here to respond to us on the biological database. So my own question, Doc, is the model structure. What is your response to the model structure that are not yet? The model structure. Where they did not use X-ray crystallography, and also they did not use electron microscope, but they modeled the structure from sequence. Has that been validated? Can one depend on such protein to do any serious docking?
Okay, I think the simple answer is yes. There's quite a number of things you can do to check the quality of the model that you built. So typically when you want to do that, you have a sequence and you want to blast that sequence and find sequences that are similar to that, what you have. in related organisms, of course. And if it's in related organisms, typically the way the protein folds are similar, and they are also guided by the nature of the amino acid. So, for example, if you have up to 50% similarity to a particular sequence, and you have the 3D structure of that particular sequence, so you are using that as a guide to build your model. Without going into so much details, there are several algorithms that can be used, but the best is all those that build based on ab initio algorithms that actually look at the property of amino acid. We know their chemistry, and we know their bonds, and we know how they have an idea of how they will orient in space. So you can use that to guess, the computer uses that to guess their position and their environment and orient them in the appropriate position. Now, of course, there can be mistakes. So that's why you do your quality check. The Ramachandra plot that I spoke about. If you put a particular amino acid in a position that is not right, you need to correct it in that model. But these days, recently, I can't remember, I think it was Google or somebody built an AI system that can actually build 3D models. And by comparing what was built from that model by that AI system to what was done experimentally, it was almost perfect according to that paper, But having said that, one of the things we do is to ensure that once you build what we call an homology model, you need to check using molecular dynamics or QMM, quantum mechanics, to see how stable the model is. If you build a model and you put it on the dynamics and it remains stable for a long time, then you can be confident that it's good for you to use for docking. If it scatters or explodes, that means some things were put in the wrong place and it will not work. So you need to go back to your algorithm and rework that until you get something. Yeah.
Yeah. Okay. Thank you. So, in absence of any other question for interaction, I think we could move to chemical database so that we can get to do hands-on, and that is why Doc have joined us so that we can apply all that has been said so far, how to get to UniProt, how to access the PDB directly, get the protein. So one of the things we must achieve today is to get to your protein of your interest, fungi protein, plasmodium protein, dengue protein for our sister working on dengue, or whatever you are working, then you get a particular protein of your interest today. So that we must achieve today as we go on. Okay, so let's quickly go to the chemical database. So we have
So we've looked at biological databases. We've seen quite a number of examples in the demo. Took you through UniProt and Protein Data Bank, which I think they're quite relevant to this session in terms of molecular docking. So I'm going to talk about chemical databases where you can find a number of chemicals that we use for this technique in terms of I'm referring to the molecular docking. And of course, so many other things that you might want to do when it comes to using molecules and compounds, small molecules. Now, chemical databases are, of course, the backbone of most of our structure-based computer-aided drug discovery, and even ligand-based, of course, computer-aided drug discovery. The central focus of cheminformatics. Okay, and what's in terms of definition, they are also very organized platform that stores digital information, which can be used to build knowledge-based models for drug discovery projects. So I just want you to take note of the fact that not only are they organized, but you also have a digital format for them. And I don't have enough time to go through the digital format, but I'll just mention a couple of them as I go through this session. All right. So on this page, I put the images from a couple of chemical databases, ranging from PubChem, which is about the largest, arguably the largest the chemical database. You have ChemSpider. You also have Campbell. I particularly like Campbell. Is well annotated, and yeah, but PubChem is also well annotated. I will show you that. I'm referring to the fact that they have good information for each chemical compound stored within the database. And you also have the drug bank, so which contains all the digital form and information related to all the FDA approved drugs. Okay, so yeah, and particularly dictionary of natural products. Although it's not a free database, but one of the most comprehensive for natural products. There are other free database that you can look at. I'll try and put a link for some of the publications that have reviewed natural product databases. And the wonderful thing about this database is they allow you to extract, so just like we're able to get the digital form of the protein and to download it into our computer, local computer, we can do the same with these databases. Allows you to get the three-dimensional structure of these compounds onto your local computer for you to do your simulation as required. So I'm just going to zone in on PubChem because I want to just show you a couple of things on PubChem, and you're going to do your hands-on exercise on this using this particular database. So it's an NIH chemical information resource, and collects data from over 620 resources for each compound. Some of them, of course, some of them might not have all the information, but yeah, for as many that are, they try to get as much information as they can for each of the compound in the database. And of course, it's free. It is claimed to be the largest, and it has a lot of information, especially related to bioactivity, that are usually extracted from scientific articles. So basically, if somebody has used that chemical and tested it against a particular disease in a particular bioactive assay, they get data from that with a link to that paper, of course, and incorporate that information into their database for that particular compound. And other annotation information, include the connection or the connection between that compound and gene and protein targets. So basically saying, for the bioactivity that have been tested, what is the potential target for this particular compound, which proteins does it interact with, and we can imagine if you know the proteins that they interact with, and from that you link that protein to a particular gene, or you link that protein to a particular disease, you can begin to also infer the kind of bioactivity to expect from each compound. So that you can just by looking at databases, there's a lot one can do in terms of research and inferring, just not only getting knowledge, but inferring some sort of conclusion, as it were, from the question that you ask of that particular database or series of databases. Okay. And it allows you to search. The PubChem platform allows you to search using keywords. If you have the keyword for that particular compound, the name, the usual name, the chemical name, anything that's even similar, allows you also to draw part of the structure or the full structure and do a search on that platform. I'm going to show you that And of course, you can also write just a small script to engage with the back end of the database and to access some compound, especially if you want to access a very large set of compound. You can just write a small script to engage with that. So there is a bar where you can type in, and if you look at, let me just show you this. So you have 3 tabs there: bioassay, compound, and substance. So under bioassay, you can also write a compound, the name of a compound. It will go under that and search for compounds that have been tested in one bioassay or the other, and of course link that each compound to that bioassay. And of course, if you have just on this tab, it gives you information about that particular compound. Although you will still see a link that if that compound has been tested in any bioassay, you'll see a link under the information that tells you where all the bioassays that have been tested for that particular compound. All right. So I kind of like this. Although I said I prefer Campbell, I like PubChem because it also has some nice tools that you can use. Bioassay tools, like I said, which is a connection to that each compound. Structure search, you can take aspects of the compound that you are querying and say, okay, can you find other compounds that have similar structure to this? And and I want to be able to expand my search space, chemical space, in terms of compounds that are very similar to this. The idea being that if they have the same substructure, they might have the same activity. Okay, so you can do a search. You can convert into a 3D conformation based on whatever format that is. Okay, I need to speak to you about the format. You can do structural clustering. Basically, this is an unsupervised learning to see, just generate a cluster tree, see which compounds are similar. You can check that using cluster analysis. I don't really want to get into that. Classification model, you can build a classification model. You can also upload compound. Remember, it's a database. They accept if you come up with a new compound that is not existing, you can upload it, and they will annotate and curate that. Then you can download specific compound. I'll use the FTP to download the whole database onto your local drive. That is also possible. All right. You can also, if you look at the search tab, you can enter the name. You can enter what we call a smile string, and you can enter what we call what I call it INCHI key for each compound. Now, compounds generally have, in order to digitize or digitalize compound, the various people have come up with various formats of representing compound. Of course, the structure of a compound that you did in your elementary chemistry. You know what a benzene ring looks like. You can physically draw that on a piece of paper. But when you want to translate that to a computer, there must be a way we can represent it such that the computer can read that information and, if necessary, give you and know that it is a benzene ring and give you that information if you require it. So there are various formats ranging from smile strings. The smile strings looks something like this. This is a smile string, and this is a key. There are other formats. There's something called SDF. It can be an SDF format. You can also have more formats. More. So it comes in that file format. Or mode 2. You have mode and you have mode 2. And you can also store them because if you look at the file you downloaded from PDB, it will have an extension called .pdb. Yeah, it should have an extension. So yeah, and the same way you can store these compounds as with an extension .pdb or .sdf.mol file. Or if it's a string, this is called a string because it's a series of characters. We call smiles, and of course, in this state, it's a specific strings of characters and numbers that forms the key. All right. So you can upload in any of these formats, but you can also upload by drawing the structure. So it has a tool that is attached to it. That's the database, and that allows one to draw the structure. So this is what it looks like. This tool, you can use this to draw the structure of the compound and upload it and do a search. I think you can do that. I'll show you something. So now in this case, the string format was imported. The smiles format was imported. Okay, and gives you, if you click on go on that, gives you identity one, similar compound one. It has one identity for that, but one identity for that particular compound. But there are quite a number of similar compounds, similar to different degrees. It will tell you the percentage similarity if you explore that. Then those that have similar substructures to your compound, it will also like that. Yeah, and some with superstructures. So different tabs, depending on what you want. And of course, you can download a CSV file. A CSV file is like an Excel file, a text file that can be opened in an Excel. Okay, and yeah, and some other information that's related to the gene, the bioassay, and the rest of them for each of the. In this case, there was just one output for this search, and I think that's the output. That's the name. Look at some of the information that you also have. Clinical trial information are available. Gives information as to which studies have been done using that particular compound, and the status of that study, the status of that such a study. Recruiting, this is the recruiting stage and the phase. This is terminated, and the trial was at phase 3. This is completed at phase 1. So it gives you all this information. And it can also give information related to pharmacology and biochemistry, and of course, the occurrence of that compound in literature. And that helps you to see how many literature have actually used that compound and for what. Was it for a bioassay or was it used as a reagent? You can also find out. That link is also there. Okay. Okay. Okay, so there is also information about the genes that are related to a particular compound. In this case, actin beta is related to this particular compound that was searched. This is the SMILES format of the compound that was put in the search there. So this particular information is related to the protein to which this compound can bind to, and the gene responsible for that protein. Okay, for the expression of that protein. So such like this. Okay. All right. So we also have information related to chemicals and the bioactivities. Okay. These are the activities: active, active. If it's not active, it will tell you, and you have a link to the paper that actually now described that bioassay or that particular activity test that was done. All right. Classification model, you can build a class something. So now in this case, your file, a CSS file is like an Excel file, a text file that can be opened in an Excel. Okay, and yeah, and some other information that's related to the gene, the bioassay, and the rest of them for each of the. In this case, there was just one output for this search, and I think that's the output. That's the name. Look at some of the information that you also have. Clinical trial information to the protein, the paper that actually now described that bioassay or that particular activity test that was done. All right, okay. More information. Sometimes, because PubChem is directly linked to the drug bank, so if the compound you search is actually in the drug bank, it gets information from drug bank and display them, and those information. In this case, it did search for a quercetin and said quercetin, yes. This is the compound CID number, but in terms, it's also a drug. And what class of drug is it? It's an experimental investigational drug. It's not been approved yet. The same thing. These are information regarded to biases. Okay. So what I did here was to list some other chemical databases, quite a number of them. You can just look at them. I try to give very short description of each one of them. And it's good to take a look so you kind of know what is the scope of each database in terms of the compounds that you can find within that data. Some of them are they are a very focused set of compounds or a library of compounds. Some are a bit more diverse and very large. Okay, at this point, I think we're going to do a demo for you on PubChem. So let me do that quickly.
Okay, yeah. That's fine. So we take a demo on the PubChem and we now resort to our hands-on.
That was a question, sir.
Okay, yeah, go ahead. Sorry, that was a question.
Yeah, I just put on the chat what smiley means. I don't understand what the person really, I'm trying to understand what the question is about. said, I don't understand what you said about smiley format.
Yeah, maybe she wants as a format.
So I cannot cover the format here, but like I said,
but I can call her up to make the question straight. Vivian, could you unmute and make your question clear so that we'll move on? Ask to unmute. Yeah. Vivian, could you unmute and ask your question?
Vivian on the line.
Thank you.
Okay. When he was trying to impute the name of the chemical, he was writing something like SFO. I tried it on my system, it couldn't come up. So I did not really get how he did that.
Okay. Okay, I'm not sure I covered that in the demo. So what I was trying to do there was to tell you that there are different formats, ways of entering the data onto PubChem. One of the formats is that smiley format that you see strings of letters. Each carbon, hydrogen, and double bonds and such like that. But there are other formats. So SDF is another format. mol is another format. Mol2 is another format. But you can leave that for now. You can read more about that. But smiley format is the one we can use as an input file for this. I'll talk more about that in the demo.
Okay, thank you.
Yeah, okay, you can move back. Thank you. Yeah, so I think we have to take the demo on this and move on.
Okay, let's continue with this session on chemical databases. So I'm just going to do a demo on how to engage with the PubChem. Once again, I'm just starting from the Google page and just typed PubChem, and I just click on that, get into the site. Okay. So the NIH National Library of Medicine, PubChem. You can read more about the database here. And remember, you can also, like I said, in most of these databases, you can submit data that you have, either for a compound that is existing there, or if you have a new compound that you want to submit, you can do that. Okay. So you can search for whatever you want, compound, small molecule. Well, they're not. I don't think they're limited to small molecules. There's some other molecules, but not proteins that are not exactly very small, but they just call them small molecules that you can use that you can search for in this database. So look at it. Let me call your attention to this at the bottom. You can use words like COVID-19 is a disease, this current pandemic, to search for compound related to that. You can use the name of the compound. You can also use the name of the protein. And you can use the formula of the compound. I've not actually used this before. Then you can use the smiles. It's a format, digital format for compounds, and the INCHI key, which is another representation of compounds. Okay, I think I'll take a look at this later. All right, so let me, for example, let's try COVID-19. Just click on that. Okay, it's telling me there are 8,498 compounds related to that condition, and there are about 15 patents. 63 of them are substances, 645 are genes, proteins. Okay, so it does contain proteins, linked to others, linked to pathway, the bioassays, and of course, literature from which these things are emerged. Okay. Interestingly, chloroquine has been linked to COVID-19, so you can see that. Ritonavir, which is a compound, an antiviral compound, has been linked to that also. So it makes sense. Savipiravir, of course, erythromycin, an antibiotic. Yeah. So, all right. So one can search with the name. We can also search with the protein. Let me see. Let me try the ACE that we used on the other side. Let's see what happens here. Okay. I didn't really see any compound related to this in terms of compound. Yeah, no. Yeah, substance is not compound. All right. So I'm trying malaria pigment. Okay. So usually, I think that's just wanted to cross check that. Usually for these chemical databases, you shouldn't be able to. It's easier to actually use the name of the chemical rather than the disease state. Okay. Perhaps because COVID-19 is it's a special case. That's why they did that link here. Okay, but easy, like I said, it's easier to put the name of the compound that you want. The protein is also, and yeah, the other formats of the compound. But I want to also focus on one other way, which sometimes you might not know the name, but you just have an idea of the structure. So, and this, I want you to be able to do this. So what I just did now, let me just repeat that. Step was to draw the structure. So I want to draw the structure. There's an icon here. You can click, takes you to an electronic chemical drawing page where you can draw a structure. So I'm just going to draw a random structure. In the hands-on exercise app, I've given you 3 compounds that I want you to draw and search for. So we're going to draw an hypothetical compound here and search for that compound. Okay, so I just pick on this benzene ring and I put it on the board. I want it attached to that ring, usually on that side. Okay, let me see. I can add a double bond here. Let me put it here. Let's see what happens. These other bonds are for stereoisomers, for compounds that have to depict chirality. I don't want to get too complex with it, but they are used to this, especially in sugar. There are compounds that have this what we call sterol chiral position, and you can use it to depict that. But let's just keep it simple for now. Let's keep it simple. Okay. So this area is for putting they are what's it called now? Pre-designed structures that you can insert. We can also like just put that here. You can put that there. You can also put this here. Okay. Now the assumption is that for this each of this position have carbon in them. So usually it doesn't show the carbon it doesn't really explicitly show the carbon. So each of these assumes their carbon, and at the end of each carbon is hydrogen. So those are not clearly shown sometimes. But you can change them if that is not the case in what you want. So for example, if what I actually wanted I had an oxygen as here, I can insert the oxygen there. Just select what you want here. If it has, for example, chlorine here, you can do that. If it has, let me see if you can put a nitrogen in there on one of the rings. Yeah, there we go. What are interesting things that are here? I'm trying to find. Of course, based on the valency of that element that you're putting in there, we could also have this another nitrogen there. If I put oxygen here, that's actually an OH that is here now. Okay, let me make it explicit. I can make it explicit by doing that, and just put the where's the hydrogen? There we go. At the end there, so there's an OH. I want to do the same thing here, so I'm going to do OH there. Because if you don't put an H here, it assumes there's a carbon here. So we need to explicitly put that there to replace the carbon that is assumed at that position. I'm going to make this, no, I want to make this a double bond. Okay, just get the carbon now into that position. Or carboxylic group into that position. All right. And you can delete them if you want to. You can just click on delete and you click on the atom you want to delete and you can get it out of that position. Okay. Now that I have the structure, now you notice that the structure, once I finish drawing the structure, you will see the smile for that structure. So this is a digital representation of this particular structure. You'll see that. If you want, you can change to a smart representation. This is a smart representation of this structure. You can also see the standard, I call it, I really don't know the pronunciation, I call it inch key. And yeah, standard inch and standard inch key representation and formula representation also for that particular structure. Okay. And give me the molecular mass for that particular structure. Okay, cool. All right, so yeah. So you don't need to copy this if you want. Once you actually copy and go back and insert on that page, but you can search directly from this chemical drawing platform on Pocket. You can just search directly and it inserted the smiles there. Identity is 0 because it doesn't exist anyway. So I can imagine. All right. So let's do something that makes more sense. I will probably find in there. Okay, let's do that. Okay, let's see. And it's just another compound on the top of my head. Okay, this one. Hopefully this will exist. Okay, there's a search for this. Oh, it still doesn't exist. Okay, so I will have to take an example from, let me take a simple example from the answer on that I gave you. Okay, let me, for that. All right, cool, we could use that one. All right, so let me use something that's similar to that. But I'm sure by now you understand the concept of what we're saying, but just let me draw a compound that no, like that. So I can actually undo. I think I can undo. Yeah, I want to put that here. All right, cool. There's a chloroquine in which position here? No. There's a chlorine rather in that position. From the structure that I'm drawing. Take a look at it again. There's an N here. There's an NH. Okay, cool. And then N here. And there's a link here. So another nitrogen. Okay. I'm gonna link that to this. All right. Okay. I think I can stop here. Hopefully I'll have something similar to this. Let me just do the search. Yeah. We're able to get one compound. Okay, there we go. So my uncompleted attempt to draw one of the compounds actually coincide with a particular compound that is in the database. All right. So if I click on that, which I want you to do for each of the compounds that I've drawn there, it takes you to the page for that compound, the compound summary. This is a summary chart. Gives you this PubChem ID, or what we call it CID, the structure of the compound. There's a 3D model also available for you to select different conformation, but let's leave that. It's a 2D representation, as a 3D representation, and formula, the synonyms in terms of the name. So you remember you have the full name here, and the full scientific name, and the molecular weight. Okay. There's a 3D conformation, I think, that should appear here. Yeah, there we go. Of the compound. You can show in different representation, ball and stick representation. It can be in wireframe representation or space fill. Yeah, so that's the case maybe. You can download the structure from here, but let's not do that for now. Okay, so on this side of the site, you'll see related information to this particular compound. We have the structure, you have the name, an identifier, so it gives all that. So if you cool down the names and gives IUPAC name, the inch and the inch key, canonical smiles, molecular formula, yeah, synonyms, chemical and physical properties, computed properties in terms of, well, some of these properties were actually predicted from computer simulation. Hydrogen bond donor, hydrogen bond acceptor, molecular weights, log P, yeah, topological polar surface area, and the rest of them. And then some related records concerning this compound. Okay. There you see that because actually I was trying to draw chloroquine, but I didn't complete the structure. So yeah, you can see compounds that are related to that. Of course, chloroquine and hydroxychloroquine came up amongst the compounds that are related to that particular structure that I drew. Similar compounds also available, similar conformers are available. You can see that. But for this compound, I'm not sure. Is there a patency? No patency. Yeah, there is a patency for this. It's a patent for that. So that's the publication. Composition and methods for the treatment of cancer. So this compound was actually tried out in this study. Okay. Yeah. Cool. So I want you to actually, in your case, maybe complete that drawing and yeah, now get more information. You will have more information specifically towards bioactivity and the rest of it. Okay, for your compound. Yeah. So please take time to engage with that. I think there are 3 compounds that I want you to draw. We will send this to you, and then I want you to be able to answer some of these questions here. There's some other activity related to another database that's very clear. You should be able to get that easily done also. But I just wanted you to explore some other databases. So that's why I inserted, I added another answer exercise that is another database beyond PubChem. Okay, beautiful. So I'll be available for questions during your answer, and to answer questions and to give insight as required. And of course, the tutor on ground will also take you through. Cool.
Yeah. Thank you so much, Dr. Sam. Yeah, so it's cool. And I think I have to stop sharing now and request everybody that is alive here to on their video, and we should interact a little. I have a few questions for Sam here. Then thereafter, we could now have a look at our hands-on and take our decisions. So please, if you are live and you are following, could you on your video now, please, and so that we could flow together. Yeah. Yeah. You see, some of these guys went to market. I was thinking they went to market. They just put this in and they went to a coca or something. Okay, I will assume that those that are.
I'm here.
Yeah, we need to see
Lillian
you to agree that you're here.
Lillian is here. Lillian has been here.
We need to see your video. That will happen from my talker.
My video is on. I have been here.
Okay, so far so good. I've personally flowed through this, and yeah, before we now begin to, because what we consolidate the study so far will be the hands-on section. So before we get to that, I wanted to make some reactions and hear from Dr. Sam. Yeah, so Sam, with what I could see that is happening in the PopChem using will I call it ChemDraw or will I call it another tool altogether? Do you think the ChemDraw is also still necessary to be installed in our system, as this seems to do everything? I don't know. You understand what I mean?
Okay, not necessarily, because you can actually get the smiles from here. So if you don't want ChemDraw, you don't have to. But it would be nice to have something on your local computer if you're going to do docking regularly.
Okay.
Something that will allow you to manipulate. And you might start with a particular compound and you want to build a series of compounds by changing a particular group, So you can keep changing that group. The R group, it allows you to change and build a library of compounds. Yeah. And you can download different formats. You cannot download, I'm not sure you can download from.
Okay.
Innocent
Mary also asked a question. So Innocent Mary, could you unmute yourself and let's get your question before I ask the rest?
No, he was talking about ChemDraw. ChemDraw is now we have become commercial and this. So I sent them Marvin Sketch. Marvin Sketch is also
it's great.
It's also okay. So I sent them Marvin Sketch because ChemDraw is now commercial or something.
Okay, cool.
Yeah, they have attracted the consign. So yeah, so I take you back. Let me check if there is a question from anybody. So Lillian Honora is asking, if one is to use the draw structure option, what informs one's consent on what to draw? Yeah, so this is a fundamental question. So, Dr. Sam?
Okay, like I said, you need to have a starting point, and you have an idea of what compound you want to, what's what I'm looking for. You have the hypothesis you want to draw. So for me, if I want to find other compounds that are similar to chloroquine, for example, that could be used for malaria. Okay, and I say, okay, I'll draw chloroquine and say search for similar compounds for that within that space. It can do that. That is the hypothesis. That is my research question. In your case, you might have a natural compound that you don't know the activity at all. It's just been isolated, but your chemistry department was able to resolve the structure of that particular compound. And you want to find out what is the bioactivity. Do we actually have a compound like this already in the database, and what are the bioactivity profile? So you can actually draw that. But most times, some of the compounds, they have the smiley formats already. So you can just input that or input the name. Yeah. So depending on what question you want to ask, it's just an option there for you. And we usually use it when you have a novel structure.
Yeah.
That doesn't have a name. Yeah. It doesn't have a name yet.
Thought. Yeah. Yeah. And also you could also use it to study synergistic actions. So because when you get compounds of similar bioactivity, you can combine them to check whether they're synergistic or antagonistic, as the case may be. Yeah, so I think it could also aid in that. Yeah, but most of the people in this training are biologists and all of that. Not many. I think he knows a few are also chemists here. So yeah, so maybe as it goes on, of course, no one man does not do research. So everybody will play his role to get something going on. Yeah, on that. So yeah, from my own table also, I would also want to ask, okay, there is nothing detailed about the plant database. Now, because for me, of course, we are working on a paper on some of the plant compounds that we have used against malaria parasites and all of these. So maybe that could make sense for me and maybe some other people here that are interested in the natural products.
Okay, so I will send a review on the platform. But from the top of my head, Supernatural is
there are several other ones we can use. It's not necessarily that it has to be that way. No, most times
Who is talking? Go ahead. Internet. Yeah, so yeah, we have the supernatural. Yeah, I think my own question is personal to me, yeah, or my own interest. So you can also put up your own question on your own interest and the one you might want to use or something like that. Dear Francisca, you the one or are you hanged? Or mute yourself. Or mute yourself. Yeah, you can go ahead. Wow, what's happening? Okay. Sam, are
No,
you
but
still here?
yeah, she's right. You can use so many, like I said, as a review paper, recently paper that reviewed natural product databases. You can actually search for it, and it's a very good review. But I think it was Tajuddin, and I can't remember his name, that reviewed it. So there are several others. It's just that dictionary of natural products is very, very comprehensive.
Dictionary.
But I mean, the others, there are a lot. And you also have them in, that's another thing, that's why I like Campbell. Campbell also stores a lot of natural products. You know, Bob Campbell also stores some. You will see some. I mean, you will find the normal quercetin, camphorol, some flavonoids, and the rest of them. Yeah, in that space.
Okay. So any other question, because we have to take a decision and I want to share a screen of our activity, but our time is fast spent. So we might need to take a decision on what to do. We make a suggestion, then we could review it all together and conclude on something. Yeah. I open our hands-on. Okay. Then and share the hands-on together with all of us. Yeah. Okay. Yeah. So this is the hands-on for those who followed, but I knew that it's clear. And maybe by tomorrow, I will also be asking us to come with our earpiece because I'm using one and I was able to hear everything. And some of us also had some, it was low volume and all of those fluctuations. Some also network could also be pulling the stuff down for you. So I think we have to arrange for earpiece and we'll still make an arrangement also to get the link across to you, but you could go through this, what I'm sharing currently, where we are to visit UniProt, get fine protease enzymes of these organisms, HIV, Plasmodium, Aspergillus. Choose one of the protein displayed on the rest, only one. Yeah, then you could describe the functions of cellular location of the protein. You locate the FASTA. So when he was teaching, all of these things has been demonstrated. Locate the FASTA sequence, find similar protein, 90, 100, 50% similarity, find the structure of the protein. If not, how will you get the structure? If the protein has PDB identifier. So we have a lot to do under biological. You download, open the file in Chimera. The exercise will continue when we now move to tomorrow, which is visualization. So chemical database, of course, okay, the focus is on small chemical compounds. So we want to work on volatile oil. So we could find the component, select one and download the mol file and SDF file. Then convert the file to mol file and PDB file format with OpenBabel. Then following what we have been taught now, I know we can easily draw this. This is, yeah, is interesting. You know, just like playing game, okay? Is to understand what is double bond, what is benzene, what is all of this and all of that. So we can practice and play around it and come up with this. And maybe when we are done, we can also search to see if we could get identity of each of the compound, describe the bioavailability, which gene has been associated, and also, though this will help you to now play through the whole thing. Now, from my time, Nigerian time should be 2:25. 2:25. So, what do you think? Could you do this now? But for the purpose of those that had issues with their, the audio and all of that, will you be so disciplined enough to, as we plan to send the recording of today, to go through the recording and now do the hands-on yourself? Then when we come tomorrow morning, we start from looking at the hands-on, the outcome of what you did. Okay? Yeah, so with that, we should be able to buy time and conclude, because our resource person in South Africa is a little bit ahead of our own time, so he might see night before us. So we are not all in Nigeria, so we need to manage. So I don't know what you think. So I want us to write in the chat what I've proposed, then we could be guided. Meanwhile, there is a question from Chisom. Does the ChemDraw have an undo button? I'm making a lot of mistakes. And but we have not guided you into ChemDraw yet. We are looking at the PopChem and the facility there we could use to draw as well. But it's good you move on and on fast. So, but first, let's respond to the proposal I've made. And I think 3 people have said if we get the record on time. So you are going to get the record immediately in your WhatsApp group. I'm going to share a link where you are going to download the record of today's lecture from the introduction to bioinformatics to where we are now. So once you get the record, you could go through it and now respond to the hands-on on your own. So 11 o'clock tomorrow, we shall now, maybe some can now demonstrate with either HIV or COVID-19 or one, then push through all of those things. Then you could use that to correct your own. Then if you have questions, you can. And I provided numbers of 2 persons that are locally located there. They could help you with the questions as you practice the hands-on. You can also put it up in the WhatsApp and they will also respond to you in the WhatsApp. So between now and tomorrow morning, we expect that you on your own, you have gone through the hands-on and you have practiced all that has been done. So that will help you to go into, just like Halimat Atanda is doing. Of course, she's practicing. So this is not a talk. We have to practice. And that is why she's encountering some mistakes and she needed to be guided. So things like that. So that makes it a workshop. At the end of the day, you would have had an idea of how to use the tool and how to navigate through the tool. So far, it's only 3 persons of 4 that have said it's okay. The rest have not said anything. And if you can say something so that maybe around 12 persons responding and that is above 50% of the persons here, then we could go ahead and take a decision and call it off for the day. So I will really want to hear from you. Okay, thank you, Godwin. Thank you, Francisca, and Chifu. Thank you. Perfect proposal. Thank you. So, okay. So is Amarachi still on this? You know, sir, it's okay. All right. So that is fine. So I'm going to work immediately. Let's get the record and do the hands-on. Perfect. Not at home. Just start doing it immediately. Chukwode. So you do it and now get home and complete it. Don't go home. You will not do it. Okay, somebody is exhausted. Okay. All right. It's nice sitting with you guys to do this training and to review. For me, it's very serious because a little bit, it's been some time I worked with this tool, but I will say to you that I'm refreshed and I hope to, engage because a lot to do on COVID now on this and look for plants. People would have worked us on compound, but we can look on plants. And get it there. So there's some, I don't know if the proposal is okay with you. So this means that 11 o'clock, you're already here and you could walk through the hands-on, then they could also confirm what they have done at home. But we shall not spend time tomorrow morning to be doing the on time with them. They would have done and maybe show us the outcome. Yeah, show us the outcome. So yeah, perfect. So what you do is to prepare a PowerPoint. As you do respond to each of the questions, you put it in the PowerPoint. So randomly, I will select people tomorrow, starting from Francisca, maybe to first present, then follow with the rest and all of that. So I've nominated you for today against tomorrow presentation. So please get ready yours whatsoever. Then tomorrow we can interact with this. And I tell you, this will help and build your confidence in molecular docking. So from my table, I think my entrance has finished. I don't know if Dr. Sam has something
Now,
to say.
I just want to lay this, the question. I think it was Chisom that asked the question. I don't know what she's using ChemDraw, or she's referring to the PubChem drawing tools. So if you look very well, you will see an undo button, and you will also see a DEL button, that's delete. So once you click on that, you can actually delete sections of the drawing that you made a mistake, and you go back. So you don't have to delete everything, but if you use Marvin Sketch, you can also use Marvin Sketch. But I don't really want you to waste time on drawing now. Just use the PubChem as a tool to draw, so you can search directly from the PubChem. So you don't get to ChemDraw or Marvin Sketch now. We'll do that later.
Okay. So any other issues, sir?
So far, so good for me. I'll be at 12. Okay, my time 12.
Okay. Okay. So thank you so much, everyone, and happy again, and to announce that we shall come to the end of this workshop so far today. And immediately I shall provide you the link to get the record for today's lecture. Please go through it again and do your hands-on. I've sent an email already for the hands-on questions, so you wish you have it in the email. And please practice, use PowerPoint to build your answers together, as we want to see what you have done tomorrow morning, 11 o'clock, before we now go into. So we're going to quickly link on from what you have done, that loaded your protein into Chimera. So visualization will commence immediately tomorrow. And of course, doctor will chair tomorrow. Yeah, fully, as I've done today. Yeah. So thank you, Innocent, and thank you, everybody, and those in the on-site. I hope the internet of the university is helping you. So thank you, and the permission to
God bless you, sir.
Yeah, end the meeting. So thank you, and bye, everybody.